Hello and welcome to the Biological Anthropology lecture series. I'm Dr. Richard Jonathan O. Tadurin. Here we explore what it means to be human, biologically, evolutionarily, and behaviorally. Let's begin our journey into the science of humanity. Welcome to Module 2, the biological basis of evolution. Here we explore cells, DNA, inheritance, and the evolutionary processes that shape all life, including humans. In this section, we examine how genes become traits through processes like transcription, translation, and gene regulation. Let's take a closer look at how DNA turns into something the body can actually use. A gene is simply a portion of DNA. On its own, it doesn't do anything. It needs to be read, copied, and translated into a functional product. That entire journey, from DNA to protein, is what we call protein synthesis. And understanding it helps us see how traits emerge at the most fundamental level. Let's look at the first major step, transcription. Imagine the nucleus as a highly protected library and DNA as a reference book you're not allowed to remove. When the cell needs a particular instruction, it doesn't take the whole book out. Instead, it makes a working copy of just the relevant page. That working copy is messenger RNA, or mRNA. Specialized enzymes unwind a section of DNA and complementary RNA bases pair with the exposed DNA bases. You might wonder why this detour through RNA is necessary. The reason is that DNA must stay protected inside the nucleus. RNA serves as the portable version of the instructions, carrying the message to where it's needed. The message encoded in mRNA isn't the final product. It's a blueprint for assembling amino acids into a protein. This brings us to translation, which happens outside the nucleus at the ribosomes. Think of ribosomes as molecular factories that read the mRNA 3 bases at a time. Each triplet, or codon, corresponds to a specific amino acid. Transfer RNA molecules, or tRNAs, act like delivery workers, bringing in the correct amino acids based on the codon sequence. As the ribosome moves along the mRNA, the amino acids link together, forming a growing chain that eventually folds into a functional protein. You might wonder how the cell ensures accuracy during this process. The complementary matching between codons and anticodons helps maintain order, and the cell has built-in proofreading mechanisms. Still, errors can occur, and even tiny changes in the sequence can influence the shape and function of a protein. Some of these changes have no effect. Others can alter biological processes in ways that become meaningful at the level of traits or even evolution. Let's look at the bigger picture for a moment. Not all proteins are simply building blocks like collagen or keratin. Many act as enzymes, hormones, or regulatory molecules that determine when other genes should be active. This means protein synthesis is not just about making physical structures. It's also about orchestrating cellular behavior. A small regulatory protein can influence entire pathways of development or metabolism. Keep in mind that every trait begins with these molecular steps: the sequence of bases, the accuracy of transcription, the precision of translation. All of these shape how an organism grows, functions, and responds to its environment. Protein synthesis is where genetic information becomes biological reality, linking the quiet world of DNA to the diversity we see in human populations. Let's spend some time with one of the most fascinating parts of this module, how genes contribute to the incredible variety we see within and between human populations. First, let's revisit what we mean by a gene. A gene is a segment of DNA that codes for a specific product, usually a protein. But that definition only scratches the surface. Genes are part of a much larger network of regulatory sequences, switches, and interactions that ultimately shape traits. When we talk about genetic variation, we're talking about differences in these segments of DNA that can lead to differences in how proteins are made or how they function. Let's look at where this variation comes from. At the simplest level, variation arises from changes in the DNA sequence. These changes called polymorphisms, can be as small as a single base substitution or as large as the insertion or deletion of entire segments. You might wonder why a small molecular change could matter. Think about how a single letter change in a sentence can shift its meaning. The same is true for DNA. A tiny alteration can modify a protein's shape or activity, which may influence a trait. One example that often appears in introductory discussions is the ABO blood group system. Here, different alleles of the same gene produce slightly different versions of an enzyme, which in turn create different surface markers on red blood cells. These markers determine blood type. It's a clear demonstration of how small genetic changes translate into observable biological differences. But it's also a reminder that many traits are shaped not by one gene acting alone, but by multiple genes working together. Now, this part is important. Structural genes, which code for proteins, are only part of the picture. Regulatory genes play a major role in determining when, where, and how strongly other genes are expressed. One powerful illustration of this is the Hox gene family. These regulatory genes don't build tissues directly. Instead, they control the sequence and pattern of development along the body's axis. They act almost like conductors in an orchestra, cueing different groups of genes at just the right time. A mutation in a regulatory gene can have profound effects because it disrupts the timing or coordination of other genes, not just a single protein. Let's think about how variation persists in populations. Genetic differences can arise through mutation, but they can also come from processes like recombination during meiosis. Recombination shuffles alleles, creating new combinations every generation. This means that even without new mutations, populations maintain a reservoir of genetic diversity. You might wonder why populations don't simply converge on a single, best genetic combination. The answer lies in the dynamic relationship between organisms and their environments. What is advantageous in one context may not be in another. Variation ensures that populations have the flexibility to respond to changing conditions. It's also worth remembering that many traits are polygenic, meaning they're influenced by multiple genes. Height, skin pigmentation, and many aspects of physiology fall into this category. For these traits, variation is continuous rather than discrete. Instead of falling into clear-cut categories, individuals fall along a spectrum shaped by the cumulative effects of many genes and environmental inputs. As we move deeper into this module, keep thinking of genes not as isolated units, but as participants in a complex interactive system. Variation at the genetic level fuels the diversity we see at the organismal level, and that diversity is the starting point for natural selection, adaptation, and long-term evolutionary change. Let's take a step back and revisit the foundations of inheritance through Mendel's work. Let's revisit why Mendel's garden peas still matter in modern genetics. Mendel wasn't thinking about DNA or chromosomes. Those discoveries came decades later, but he was able to uncover patterns that revealed how traits are transmitted from one generation to the next. His observations gave us the basic principles that help explain why some traits appear, why some skip generations, and why variation persists. Let's look at the first key idea, the law of segregation. Mendel noticed that traits don't blend. Instead, the units of inheritance, what we now call alleles, separate during the formation of gametes. You might wonder how this fits with what we learned about meiosis. During meiosis, homologous chromosomes separate, and each gamete receives only one allele for each gene. That cellular process is the biological mechanism underlying Mendel's pattern. What appears as a simple 3-to-1 ratio in his pea plants reflects chance combinations of alleles during fertilization. Mendel also discovered the law of independent assortment, which tells us that different genes are inherited independently if they are located on different chromosomes. In cellular terms, this independence comes from the way chromosome pairs line up and separate during meiosis, but not all genes assort independently. Genes located close together on the same chromosome can be inherited as a set, something Mendel couldn't have known. This is where modern genetics expands beyond his early insights. Let's think about how these principles connect to real human traits. Many traits follow Mendelian patterns, like certain blood groups or some inherited disorders. But most human traits do not fit neatly into simple dominant recessive categories. Height, skin pigmentation, and many physiological traits are polygenic, shaped by interactions among multiple genes. Others are influenced by regulatory genes, which control when and how structural genes are expressed. Environmental factors also play a major role, adding another layer of complexity. So, while Mendel gives us the starting framework, the real world of genetics is richer and more dynamic. Mendelian principles help us understand the basics, but they are only the beginning of how genes, environments, and development work together to produce the diversity we study in biological anthropology. First, let's revisit what we accomplished in this section. We followed the journey from DNA to RNA to protein and then stepped back to see how differences in genes and differences in their regulation shape the traits we observe in human populations. You might wonder why tracing this path matters in a field that often looks at fossils, skeletons, or large-scale patterns of variation. The answer is that every visible trait, every adaptation, every inherited condition begins with these molecular events. Let's look at the bigger idea that ties everything together. Genes don't act in isolation. Transcription and translation might seem like straightforward steps, but the cell constantly regulates which genes are active, how strongly they're expressed, and when their products are needed. Variation can enter the system at multiple levels, through changes in DNA sequence, through recombination, or through differences in gene regulation. That variation is what allows populations to remain flexible and responsive to changing environments. When we think about Mendelian principles, it becomes clear that simple dominant and recessive patterns explain only part of the story. Many traits are polygenic, shaped by the combined influence of multiple genes. Others reflect the action of regulatory networks that control development or physiology, and all of these traits interact with environmental factors, adding another layer of complexity. As we close this section, keep in mind that understanding how genes become traits gives us the bridge between molecular biology and human evolution. It's where biological instructions become biological diversity, and where the groundwork for natural selection is set. Thank you for joining this lecture. I'm Dr. Richard Jonathan O. Tadurin. I hope this session deepened your understanding of biological anthropology. Keep exploring what it means to be human. See you soon.